So, let's go. Number 1. Fatal familial insomnia. Imagine a body that simply forgets how to sleep. Not a bad night. Not a month of restlessness. A permanent, total, irreversible loss of sleep that ends only in death. This is the most disturbing illness in all of medicine, and it is written into the genes of a small number of families who have carried it for centuries. The cause is a single change in a gene called PRNP. At position 178, one letter of the genetic code flips. When that flip sits beside a specific version of another position, called codon 129, the result is fatal familial insomnia. Change that second position, and the very same first mutation produces a different disease entirely. It is a genetic coin flip, and the family carrying it has no say in the toss. The condition was formally described in 1986 by the Italian physicians Elio Lugaresi and Rossella Midori. Their patient came from a family near Venice whose relatives had died, generation after generation, of a strange sleeplessness no one could name. A researcher named Ignazio Reuter helped trace the family tree back through the centuries. What they found was a mutation that does something almost unbelievable. It turns the brain's own prion protein into a misfolded form that cannot be broken down. This misshapen protein piles up in the thalamus, the brain's relay station and the gatekeeper of sleep. As the thalamus degrades, the gate jams shut, and sleep never comes again. The disease moves through 4 stages. First, worsening insomnia arrives with panic and strange phobias. Then come hallucinations and agitation. In the 3rd stage, sleep vanishes completely, and the body wastes away. In the 4th, dementia and silence precede death. Patients enter a state doctors call agrypnia excitata, a sleepless overdrive of the nervous system, with racing heart, soaring blood pressure, drenching sweat, and fever. Sleep studies show the electrical signatures of rest simply disappearing. Median survival after symptoms begin is about 18 months. Why can modern medicine not stop it? Consider 3 explanations. The mainstream view holds that a misfolded protein offers no genetic material to target with drugs. A second view argues the thalamic damage is already irreversible by the time symptoms appear. A third points to how rare and late diagnosed the disease is, leaving almost no window to intervene. 2 researchers, Sonia Valab and Eric Minikel, learned that Sonia carried the mutation and changed careers entirely to hunt for a cure using gene silencing therapy. Their search continues. From a protein that cannot be drugged, we turn to an organism that eats the brain outright. Number 2. Naegleria Fowleri. It lives in warm, fresh water almost everywhere, and most of the time it eats bacteria and bothers no one. But under the wrong conditions, this single-celled amoeba becomes one of the most lethal organisms on Earth. It was first identified as a human killer in 1965 by the Australian researchers Malcolm Fowler and Rodney Carter. The disease it causes has a fittingly clinical name, primary amoebic meningoencephalitis, and a horrifying reality behind it. The amoeba destroys the brain from the inside. The route it takes is the reason it is so deadly. When contaminated water is forced up the nose, the amoeba latches onto the olfactory nerve, the nerve of smell. It follows that nerve upward, passes through a sieve-like plate of bone called the cribriform plate, and arrives at the base of the brain. There it begins to feed. It uses structures shaped like tiny suction cups, called food cups, to tear away and swallow living brain cells one piece at a time. This organism thrives in heat, growing happily at temperatures up to about 45 degrees Celsius, which is why warm lakes, hot springs, and poorly treated pools are the danger zones. The numbers are almost hopeless. The fatality rate is above 97%. Of the hundreds of documented cases in the United States, only a small handful of people have ever survived. The timeline is brutally fast. After an incubation of 1 to 9 days, symptoms begin with a crushing frontal headache, fever, and a stiff neck, and death usually follows within about 5 days. You cannot catch it by drinking water. The nose is the only door. Tragically, several deaths have followed the use of ordinary tap water in nasal rinsing devices and in ritual washing. One survivor changed the conversation. In 2013, a young girl named Kali Hardig survived after being treated with an experimental drug called miltefosine, combined with deliberate cooling of her body to protect the brain. Miltefosine began life as an anti-cancer and anti-parasitic drug, and it remains the leading experimental therapy, though it is far from a reliable cure. The amoeba itself belongs to an ancient lineage of eukaryotes that predates humanity by hundreds of millions of years. Why does it kill so completely? 3 theories compete. One blames the blood-brain barrier for keeping drugs out. Another blames sheer speed. A third blames misdiagnosis, since early symptoms mimic ordinary meningitis. From a killer that liquefies the brain, we move to a worm you can watch crossing your own eye. Number 3. Loa Loa. There are few sights in medicine more unsettling than a living worm swimming slowly across the surface of a human eye. That is the calling card of Loa Loa, a parasitic worm of the rainforests of West and Central Africa. The infection it causes is called loiasis, and it begins with the bite of a deerfly of the genus Chrysops, sometimes called the red fly or mango fly, which breeds in the muddy streams of the forest. When the fly bites, it delivers immature worms into the skin. Those worms grow into adults that migrate through the tissue just beneath the skin. The females can reach about 7 centimeters in length. As they travel, they sometimes cross the clear surface of the eye, in full view, before disappearing back beneath the skin. The offspring of these worms, called microfilariae, circulate in the blood on a daytime schedule that matches exactly when the day-active flies are biting. It is a parasite finely tuned to its messenger. The migration also produces sudden temporary swellings called Calabar swellings, named for the town in Nigeria where they were commonly recorded. European physicians documented worms in the eyes of enslaved West Africans as early as 1770. Here lies the cruel heart of the disease. There is no vaccine, and there is no safe way to treat entire populations at once. The drugs that kill the microfilariae, diethylcarbamazine and ivermectin, work well in most people, but in patients carrying enormous numbers of the parasite, killing them all at once can trigger a fatal brain reaction called encephalopathy. The best medicine becomes the deadliest risk in exactly the sickest patients. This danger collides with the great campaigns to fight river blindness, which use the same drug across the same forests. To solve it, a team including the laboratory of Daniel Fletcher built a smartphone-based device called the Lowscope, which counts the worms in a single drop of blood before treatment begins. An estimated 10 million people or more carry this parasite. Because it rarely kills directly, it has long been neglected. Yet filarial worms have co-evolved with primates for tens of millions of years, making this an ancient partnership between parasite and host. Removing the visible eye worm is a minor procedure, but it does not clear the body. Why does loiasis resist a clean solution? 3 ideas dominate. One points to the overwhelming parasite load. A second points to drug toxicity. A third points to a vector that breeds across a forest too vast to control. From a parasite of the trees, we turn to a virus riding ticks out of the open steppe. Number 4. Crimean-Congo hemorrhagic fever. This is a hemorrhagic fever with an ancient partnership between tick and virus, and it is now spreading into new lands. The disease is caused by the Crimean-Congo hemorrhagic fever virus, a member of a family of tick-borne viruses. Its double name comes from 2 moments in history. It was first described among Soviet troops in Crimea in 1944, where it was called Crimean hemorrhagic fever. Later, in 1956, the same virus was isolated in the Congo, and the two names were fused into one. The key player is the Loa Loa tick, which serves as both the carrier and the reservoir of the virus. People become infected in several ways. A tick may bite them. They may crush an infected tick with bare fingers. Or they may handle the blood and tissue of infected livestock during slaughter. This last route makes farmers, herders, and slaughterhouse workers especially vulnerable. The virus also spreads inside hospitals, and health workers have died after contact with patients' blood. After an incubation of 1 to about 13 days, the illness begins abruptly with fever, aching muscles, and headache. Then the bleeding starts, with pinpoint spots, deep bruising, and hemorrhage from the gut and mucous membranes. The fatality rate runs from roughly 10 to 40%. The virus is endemic across a wide belt, from Africa through the Balkans and the Middle East, and into Asia, tracking exactly where its tick lives. And that range is growing. A warming climate and the movement of livestock are pushing the Loa Loa tick into parts of Western Europe where it was never established before. Migratory birds carry immature ticks across whole continents, seeding new pockets of infection. Amplifying hosts, such as ostriches and hares, help the virus multiply in the wild. Modern medicine has no strong answer. There is no broadly licensed vaccine, though a locally produced vaccine has been used in Bulgaria without wide validation. The antiviral ribavirin is used, but its benefit is debated and unproven. The virus is so dangerous that it must be handled in maximum containment laboratories, and the World Health Organization lists it as a priority pathogen with epidemic potential. Why can it not be controlled? 3 explanations stand out. One blames the complex ecology of the tick. A second blames the many animals that host the virus. A third blames the sheer difficulty of building a safe, effective vaccine. From a virus carried by ticks, we move to one that drips from fruit bats into the sap of a palm tree. Number 5. Nipah Virus. This virus emerged from the collision of bats, pigs, and people, and it kills a staggering fraction of those it infects. Nipah is a member of the Nipah Virus group, a close relative of the Hendra virus. It takes its name from Sungai Nipah, the Malaysian village at the center of the first recognized outbreak in 1998 and 1999. The Malaysian virologist Kau Bing Chua isolated the virus in 1999, identifying a completely new threat. The natural home of the virus is the fruit bat, the great flying fox of the genus Herpes. In Malaysia, the virus passed from bats to pigs, and then from pigs to the farmers who raised them. More than 100 people died, and to stop the outbreak, authorities culled roughly 1 million pigs. In Bangladesh and India, the virus takes a different path. There, people drink raw date palm sap collected overnight, and bats visiting the collection pots contaminate that sap with saliva and urine. The virus also spreads directly from person to person, especially among caregivers and inside hospitals. The lethality is frightening. Depending on the outbreak, the fatality rate has ranged from about 40% up to 75% or higher. After an incubation of 4 to 14 days, the illness can bring fever, headache, drowsiness, and disorientation, sometimes with severe respiratory distress, progressing to swelling of the brain and coma. Some survivors suffer a relapse of brain inflammation months or even years later. There is no licensed treatment and no licensed vaccine. A monoclonal antibody known as M100 2.4 has been given on a compassionate basis, but it is not a proven cure. The World Health Organization lists Nipah as a priority pathogen with pandemic potential, and it is handled only in the highest containment laboratories. In 2018, an outbreak in Kerala, India, was contained through rapid contact tracing, a reminder that speed and organization can still hold the line. Bat Nipah viruses are ancient, with their divergence placed deep in evolutionary time by molecular clocks. Yet the risk to humans is thoroughly modern, driven by deforestation and the spread of orchards into bat territory. Why does Nipah keep spilling over? 3 theories converge. One blames habitat loss, forcing bats and people together. A second blames contamination of the food chain. A third blames the virus's own adaptability. From a bat virus that jumped through pigs, we turn to its filovirus cousin, first unleashed inside a European laboratory. Number 6. Marburg Virus. Marburg is the older cousin of Ebola, and its story began not in a remote village, but inside the laboratories of a wealthy European city. In 1967, lab workers in the German cities of Marburg and Frankfurt, and in Belgrade, fell gravely ill after handling African green monkeys imported from Uganda. 31 people were infected, and 7 died. The disease was new, and it took the name of the city where it first appeared. Marburg is a filovirus, part of the same family as Ebola, and it causes a severe hemorrhagic fever. The natural reservoir of the virus is the Egyptian rosette, a fruit bat that roosts in caves and mines. This is why so many outbreaks are linked to underground spaces. A long outbreak in the Democratic Republic of the Congo between 1998 and 2000 was traced to a gold mine full of bats. The fatality rate varies enormously, from about 24% in that first laboratory outbreak to 80 or 90% in later African epidemics. The outbreak in Angola in 2004 and 2005 killed roughly 90% of more than 250 people infected. Under the electron microscope, the virus is unmistakable, a long thread that sometimes curls into hooks and rings. After an incubation of 2 to 21 days, illness begins abruptly with high fever, severe headache, and deep malaise. By the second week, the body may bleed from many sites, and the patient can slide into shock and multi-organ failure. There is no approved vaccine and no approved antiviral, though experimental vaccines and antibody treatments are being developed, many of them adapted from the fight against Ebola. Miners and cave tourists face elevated risk from exposure to bat roosts. Recent years have brought fresh outbreaks in Equatorial Guinea and Tanzania in 2023 and in Rwanda in 2024, proof that the virus keeps spilling over. A related species called raven virus adds to the threat. Deep within many mammal genomes, scientists find endogenous filovirus-like elements, genetic fossils that prove this viral family is extraordinarily ancient. Why does lethality swing so widely between outbreaks? 3 explanations compete. One points to differences in viral strain. A second points to the health and genetics of the host population. A third points to how quickly good medical care arrives. From a virus that kills within days, we turn to one that hides in the body for a lifetime and quietly turns cells cancerous. Number 7. Kaposi's Sarcoma Herpesvirus. Most of the viruses on this list announce themselves violently. This one does the opposite. It slips into the body, hides for life, and in many people never causes a symptom at all, until the day it paints the skin with tumors. Known formally as human herpes virus 8, it was discovered in 1994 by the researchers Yuan Chang and Patrick Moore. It is the cause of Kaposi's sarcoma, a cancer of the cells that line blood vessels, which produces distinctive purple and brown lesions on the skin and inside the body. The cancer itself was described much earlier, in 1872, by the Hungarian dermatologist Moritz Kaposi. For more than a century, its cause remained a mystery. The virus also drives 2 other serious conditions, primary effusion lymphoma and multicentric Castleman disease. Like all herpes viruses, it establishes lifelong latency, meaning it hides permanently inside cells and cannot be cleared. It carries a toolkit of pirated genes that mimic human genes, including a viral version of a growth control protein and a viral copy of an immune signal called interleukin-6, which help push infected cells to multiply. The virus became widely visible during the AIDS epidemic because a weakened immune system allows Kaposi's sarcoma to grow aggressively. Its prevalence varies sharply by region, high across sub-Saharan Africa and parts of the Mediterranean, and lower in northern Europe and the United States. It spreads through saliva, through sexual contact, and through organ transplantation. Its family tree tracks ancient human migrations, a sign that it has traveled with our species for a very long time. Endemic Kaposi's sarcoma existed in Africa long before HIV appeared, proof of the virus's deep history. There is no cure that clears the hidden virus. Treatment targets the cancers it causes, not the infection itself. Restoring the immune system, for example through antiretroviral therapy in HIV patients, can cause the lesions to fade. A viral protein called LANA anchors the viral genome to the host's own chromosomes, so that every time a cell divides, the virus is copied along with it. This is one of only 7 viruses known to cause cancer in humans. Why can it not be cured? 3 reasons dominate. One is the reservoir of latent virus. A second is that anchoring trick that ties the virus to our chromosomes. A third is that clearing it depends on the immune system, not on any drug. From a virus that needs only us, we turn to one that cannot survive without another virus at its side. Number 8. Hepatitis D, the Delta agent. This is the smallest virus known to infect humans, and it is also the one that turns hepatitis into its deadliest form. It was discovered in 1977 by the Italian researcher Mario Rizzetto, working in Turin. What makes it so strange is that it cannot live on its own. Hepatitis D is a satellite virus, a parasite that needs another parasite. To assemble itself and infect liver cells, it must borrow the outer coat of the hepatitis B virus. Without hepatitis B, hepatitis D is helpless. Its genome is a tiny circle of RNA, only about 1,700 building blocks long. It performs chemical tricks found almost nowhere else in human viruses, using a self-cutting piece of RNA called a ribozyme and hijacking the host's own machinery to copy itself. There are 2 ways to catch it. In co-infection, a person is hit by hepatitis B and D at the same time. In superinfection, hepatitis D lands on top of an existing chronic hepatitis B infection, and this is the more dangerous scenario. Either way, the combination produces the most severe form of viral hepatitis, accelerating the path to cirrhosis and liver cancer far faster than hepatitis B alone. Between 12 and 20 million people worldwide are estimated to be infected, concentrated in pockets around the Mediterranean, in Central Asia, in West Africa, and in the Amazon. These clusters reflect long-standing local circulation of the virus. The standard drugs used against hepatitis B fail against hepatitis D because they do not stop production of the surface coat that Delta needs. For years, the only option was pegylated interferon, with low cure rates and heavy side effects. A newer drug called bulevirtide, approved in Europe in 2020, blocks the virus from entering liver cells, but it suppresses the infection rather than reliably curing it. There is a hopeful twist. Because hepatitis D depends entirely on hepatitis B, vaccinating against hepatitis B also prevents hepatitis D. The origin of the Delta agent is debated, and its ribozyme resembles those of plant-infecting agents called viroids, hinting at an ancient lineage reaching back toward the RNA world. At least 8 genetic types exist, with different severity in different regions. Why does it resist cure? 3 theories stand out. One is its dependence on the hepatitis B coat. A second is hepatitis B genetic material woven into liver cells. A third is the resilience of that self-cutting RNA. From a rare parasite of a parasite, we turn to a virus that almost everyone on Earth already carries. Number 9. Cytomegalovirus. This may be the most successful virus you have never heard of. It infects a majority of all adults on the planet. and most of them will never know. Known formally as human herpesvirus 5, it takes its name from the swollen cells it creates, which under the microscope show a distinctive mark that pathologists call the owl's eye. The enlarged cells were noticed in the early 20th century, and the virus itself was isolated in the 1950s by teams including Thomas Weller. Like its herpesvirus relatives, it establishes lifelong latency, hiding permanently in the body's cells. Its reach is enormous. Depending on the population, somewhere between 50 and 80% of adults carry it, and the figure runs higher in the developing world. For most healthy people, it is harmless, but for two groups, it is devastating. The first is the unborn. Cytomegalovirus is the most common infectious cause of birth defects in the world. When it passes from mother to baby during pregnancy, it can cause hearing loss, an abnormally small head, developmental delay, and vision problems. Roughly 1 in 200 babies is born already infected, though most show no symptoms at birth. The second group is people with weakened immune systems, such as transplant recipients and those with advanced HIV, in whom the virus can attack the eyes, the gut, and the lungs, and can kill. The virus spreads through saliva, urine, breast milk, sexual contact, blood transfusion, and transplanted organs. Several antiviral drugs, including ganciclovir, valganciclovir, foscarnet, and letermovir, can suppress it, but none can clear the latent virus from the body. Worse, the virus can develop resistance to these drugs, especially in transplant patients on long courses of treatment. There is no licensed vaccine, although developing one has been a priority for decades, and an advanced messenger RNA candidate is now in late-stage trials. Cytomegalovirus has the largest genome of all the human herpes viruses, around 235,000 base pairs, and it devotes a remarkable number of those genes to hiding from the immune system. Its lineage tracks the evolution of primates over deep time. Research has even linked it to the aging of the immune system in the elderly. Why is it incurable? 3 reasons recur. One is the reservoir of latent virus in the body's cells. A second is its arsenal of immune evasion genes. A third is the simple permanence of latency itself. From a virus carried by billions, we turn to one that strikes only a handful of people a year, yet kills a third of them. Number 10. Eastern Equine Encephalitis. It is rare enough that most people have never heard of it, and deadly enough that a single bad season can shut down entire towns. Known by the nickname Triple E, it is caused by a virus in the alpha virus group. In nature, the virus cycles quietly between songbirds and a specific swamp mosquito called Culiceta melanura, which lives in freshwater hardwood swamps and rarely bites people. The danger comes when other mosquitoes, feeding on both birds and mammals, act as bridges and carry the virus to humans and horses. The disease was first recognized in horses in the northeastern United States, with a major equine outbreak recorded in 1831, and the virus was isolated in 1933. Human cases are rare, averaging only a handful across the United States each year. But when the virus invades the nervous system, it is among the most lethal of all mosquito-borne diseases. The fatality rate for that severe form is roughly 30%, and about half of those who survive are left with lasting neurological damage, including seizures, paralysis, and cognitive impairment. After an incubation of about 4 to 10 days, symptoms can escalate from high fever and headache to vomiting, seizures, and coma. There is no specific antiviral treatment and no licensed vaccine for humans, although a vaccine does exist for horses. Cases cluster in the eastern and Gulf Coast United States, with hotspots in New England, Florida, and the Great Lakes region. In 2019, an unusual surge produced dozens of cases and several deaths, prompting towns to spray for mosquitoes from the air and to impose evening curfews. Related strains circulate in Central and South America. Warm, wet conditions that boost mosquito populations raise the risk, and horses, which suffer high mortality, often fall ill before humans do, serving as an early warning. The virus is even classified as a select agent because of its potential to be spread through the air and the lack of any countermeasure. The swamp cycle that sustains it, between birds and mosquitoes, is ancient, reaching back long before human settlement. Why is it so dangerous and so untreatable? 3 theories stand out. One points to the virus's raw ability to attack nerve tissue. A second points to its rarity, which slows the development of treatments. A third points to the simple fact that no drug or vaccine exists. 10 diseases, 10 different mechanisms, and yet one shared reason medicine keeps losing. Look back across these 10, and a pattern emerges. Every one of them is far older than human civilization. The herpesviruses, Cytomegalovirus, and the Kaposi Sarcoma virus belong to a family that has co-evolved with backboned animals for hundreds of millions of years. The filarial worm behind loiasis has parasitized primates for tens of millions of years. The brain-eating amoeba belongs to an ancient branch of single-celled life that was thriving in soil and water long before the first humans walked. The viruses hide their age in our own genes. Filoviruses like Marburg have left endogenous elements, genetic fossils, scattered through the genomes of many mammals. The delta agent behind hepatitis D carries a self-cutting piece of RNA that resembles the machinery of plant viroids, pointing back toward the ancient RNA world at the dawn of life. And prions, the misfolded proteins behind fatal familial insomnia, represent a form of biological inheritance older than any genome record, a way of passing on shape rather than code. The bird and mosquito cycle behind Eastern equine encephalitis and the bat reservoirs behind Nipah and Marburg all predate human settlement by ages. This is why the word prehistoric fits. These agents are older than writing, older than farming, older than cities. The tool scientists use to date them is the molecular clock, which measures how fast genetic sequences change and works backward to estimate ancestry. There is an important distinction to keep in mind. An organism can be ancient even when the disease it causes was only recently recognized. Nipah was named in 1999, but the virus is old. Again and again, the story is the same. Humans are the newcomers, stumbling into biological loops that were running long before we arrived. That raises the real question. Why does being ancient make these diseases so hard to cure? The answers cluster around a few stubborn themes. The first is latency. The herpesviruses do not fight and leave. They hide, tucking their genetic material inside our cells where drugs cannot reach without destroying the cell itself. You can suppress them, but you cannot evict them. The second theme is the strange chemistry of prions. A misfolded protein carries no genetic material to target, so the entire toolkit of modern antiviral and antibiotic medicine simply has nothing to grab. The third theme is the body's own defenses working against us. The blood-brain barrier, which shields the brain from harm, also shields infections, like the brain-eating amoeba from treatment. The fourth theme is the treatment paradox, seen most clearly in loiasis, where the drugs that kill the parasite can kill the patient when the parasite load is too high. The fifth is dependence and disguise, like hepatitis D surviving by hiding inside the coat of hepatitis B, and cytomegalovirus spending much of its large genome on genes that blind the immune system. There are practical reasons too. Diseases like Marburg, Nipah, and Eastern equine encephalitis are dangerous and relatively rare, which means they must be studied in maximum containment laboratories and attract limited commercial investment. Many are neglected tropical diseases, affecting the world's poorest people, where the money for research runs thin. Building vaccines against rapidly changing RNA viruses is genuinely hard, and there is a difference that matters enormously, the difference between suppression and cure, between control and eradication. For most of these 10, medicine can manage, but it cannot win outright. And now, a set of modern forces is making the old enemies bolder. The ancient agents are not staying put. Warming waters are extending the range of the brain-eating amoeba northward, with cases now appearing in states once thought too cool for it. The Hyaloma tick that carries Crimean-Congo hemorrhagic fever is expanding into Western Europe as the climate warms and livestock move across borders. Deforestation pushes fruit bats into closer contact with people and their orchards, raising the odds of a Nipah or Marburg spillover with every acre cleared. Longer, warmer, wetter seasons mean more mosquitoes, and more mosquitoes mean more chances for eastern equine encephalitis to reach people. Migratory birds ferry infected ticks across whole continents. Urban growth presses into the habitats of wild reservoirs, and global travel can carry a rare infection across the world in a single day. The surge of eastern equine encephalitis in 2019 and the string of recent Marburg outbreaks in Equatorial Guinea, Tanzania, and Rwanda read like warning flares. The pattern is a compounding one. Old pathogens, perfected over millions of years, are meeting new conditions that widen their reach. Surveillance, meanwhile, remains thin in exactly the regions where these diseases are most entrenched, so outbreaks can smolder before anyone notices. It would be easy to end on despair, but that would miss half the story, because the same science that dates these diseases to deep time is also, slowly, learning how to fight them. The progress is real, even where cures remain out of reach. For prion diseases like fatal familial insomnia, researchers are developing gene-silencing therapies called antisense oligonucleotides, designed to shut down production of the dangerous protein before it can misfold. For the brain-eating amoeba, the combination of miltefosine and deliberate body cooling has produced a small but growing number of survivors. For loiasis, the Loa now lets doctors measure the parasite load in a drop of blood, making treatment far safer. For Nipah and Marburg, monoclonal antibodies, laboratory-made immune proteins, are being tested as emergency treatments, and filovirus vaccine platforms built during the fight against Ebola are being adapted to Marburg. For hepatitis D, bulevirtide has become the first drug designed specifically to block the virus from entering liver cells. For Cytomegalovirus, an advanced messenger RNA vaccine is now in late-stage trials, carrying real hope of preventing the birth defects it causes. Faster genetic tests and point-of-care diagnostics are helping catch outbreaks earlier, and the World Health Organization's priority pathogen lists are steering money toward the most dangerous gaps. None of this amounts to a clean victory. These remain diseases that medicine can often blunt but rarely erase. Yet incremental control still saves lives, and every survivor of a 97% fatal infection represents a genuine breakthrough. The frontier now is latency and misfolding, the two hardest problems, the reasons these ancient agents cling so tightly. Which leaves one last question worth sitting with. What do these 10 diseases, older than our species, ultimately teach us? The clearest lesson is an uncomfortable one. Across this entire list, age and incurability travel together. The oldest agents, the ones woven most deeply into the fabric of life, are precisely the ones modern medicine struggles hardest to defeat. That is not a coincidence. It is the result of millions of years of adaptation, of pathogens learning to hide, to disguise, and to persist inside the very machinery of their hosts. It punctures a comfortable illusion, the idea that modern medicine has conquered infection. Antibiotics and vaccines transformed the world, but latency and misfolding remind us that biology is stubborn and that some problems do not yield to a single clever drug. For many of these diseases, the smartest strategy is not cure, but prevention. Stopping the tick bite, the nasal rinse with untreated water, the contaminated palm sap, the mosquito at dusk. Each illness teaches a slightly different lesson about the limits of what we can do. There is an ethical weight here too. Several of these are neglected diseases of the poor, and the gap in research funding is a choice, not a fact of nature. Humility, serves us better than triumphalism. Surveillance, prevention, and steady research will do more than any promise of a final conquest. These 10 agents also hold up a mirror, for the fossils of ancient viruses sit inside our own genomes, and some of our genes were borrowed from them. The future they point toward is not eradication, but coexistence, a wary sharing of the planet with adversaries far older than we are. They remain unbeaten, and they are still, patiently, teaching us how little we have truly mastered.